Hello everyone, myself Chaudhary Govind Radheshyam, Assistant Professor at the Department of Zoology, Maharaj Singh College, Saharanpur, Uttar Pradesh. I have prepared an assignment teaching a concept. The topic which I have chosen here is polymerase chain reaction. So before proceeding our lecture, here we find an image. This image is indicating something and something is happening in this image. So what are these things? So these are actually steps of the PCR, such as denaturation, annealing, and extension. And what are the importance of these steps? What are the principles behind these steps? All of these things we will discuss in more detail in this lecture. So let's start our lecture, polymerase chain reaction. These are the important points which we will discuss in this chapter. First, we will discuss general introduction about PCR. What are the reagents which are required in the polymerase chain reaction? What are different steps in the PCR? What are the principles behind different steps in the PCR? We will discuss in this section. And what are the properties of primers? What are the how to design a good primer? What is a bad primer? We will discuss in this section. And at last, we will discuss what are the application of PCR in the field of science. Let's begin with our introduction part about polymerase chain reaction. The first questions which comes in our mind is what is a PCR? So PCR is basically a thermocycler machine. In this machine, a different gradient of temperature is utilized to amplify a particular segment of a DNA in a millions and billion of a copy. So in this polymerase chain reaction, the main and the most important component is the temperature. So each temperature is responsible for each steps in the PCR. So basically in the polymerase chain reaction there are three steps, that is denaturation, and second step is annealing, and third step is extension. And all of these steps are regulated by different temperature gradient. Each temperature gradient is responsible for each steps. Suppose for denaturation, 94 degree temperature are required. For annealing to occur, is 50 to 65 degree temperatures are responsible, and for extension process, 72 degree temperature is responsible. And all of this temperature are repeated in a cyclic manner so that a particular region of a DNA get amplified in a millions and a billions of a copy. So to understand in simple way, I have chosen one animated video so that you can understand very easily. So I will play this video. Suppose it is a region of interest, and if you want to amplify this segment of a DNA, so how this happen? First step is denaturation, 94 is annealing, and then third step is extension. So again here denaturation and then annealing occur at 50 to 65 degrees Celsius, where the primer get annealed to its complementary segment of a DNA. And the third step is extension, and extension take place at the temperature 72 to 75 degrees Celsius. You will see here. So by this way, a single segment of a DNA gets amplified in a millions and a billions of a copy. So at last we have a billions of copy of a particular segment of a DNA. So these are the basic steps of the PCR: denaturation, annealing, and extension, which are regulated by specific temperature. So now we will come what are the basic requirement of PCR. First there is itself DNA, and then buffer are required, which are responsible for creating an environment for maintaining an environment for polymerization, and then building blocks, nucleotides is responsible, which utilized by DNA polymerase to extend the primer. And primer is itself responsible for specific polymerization. Suppose a particular segment of a DNA, if you want to amplify a particular segment of a DNA, then we will design a specific primer so that only that segment of DNA gets replicated, not the whole DNA gets replicated. So now we will discuss about the primer, how to design a good primer so that we get only desired specific, so that the specific segment of DNA only gets amplified, not the other region of DNA gets amplified. So how to choose a good primer? So here I have discussed some of the important point to design and how to design a good primer. First is choosing the primer length. What should be the primer length? It should be between 18 to 25 nucleotides. Suppose we are choosing a primer length of very less base. Suppose we are choosing 5 base, 4 base. Then what will happen? In that situation, we will find that we will get not a single band, we will get multiple band of undesired product. So this will happen. So we have to choose a optimum nucleotide length. And the second important thing is melting temperature. The melting temperature between the primer pair should not exceed 5 degrees Celsius. Suppose the forward primer is TM of forward primer is 55 and TM of a reverse primer is 65. So difference is 10 degrees Celsius. And suppose we'll set the annealing temperature at the 55, then only the forward will be extended, forward will be replicated, not the reverse. So we will get the product very less product. So we have to keep on mind that product. So the TM value should not exceed more than 5 degrees Celsius between the primer pair. Third is DC content. We know that extension occur at 75 degrees Celsius, and at the high temperature, if our primer is not annealed in a well manner with the DNA template, and suppose at the temperature the primer get peel off, it falls from the complementary DNA, then how it will replicate? So we use 40 to 65% GC content, so that the bond between G and C is triple bond and it is quite stable at the higher temperature. So we use GC content 40 to 60 degrees Celsius, 40 to 60%. And one more important thing is that primers should be complementary to DNA strand. They should not be complementary to each other. The forward primer should not complementary to reverse primer. If suppose this situation take place, then what will happen? We will just see with the help of this video. Suppose it is forward primer and it is reverse primer. It should complementary to this, it should complementary to this strand. It should not be complementary to each other. And suppose if these two are complementary to each other, then in this situation we find that two products. One is a desired product of this segment of a DNA and another is undesired product. We call it as a primer dimer. So how we will recognize this primer dimer? So in the gel we find that undesired band here, this. So it is undesired product. So we have to keep in mind before designing the primer. And third is the self complementary. The primer should not be self complementary to itself. If suppose it is self complementary, then it will not bind to the specific segment of a DNA and we will not get any product. And at last we will discuss about the application of the PCR. It is used in genotyping, it is used in cloning, it is also used in mutation detection, whether it is a loss of function mutation, whether it is a null mutation, whether it is a hypermutation, we will see with the help of the PCR. Using the specific primer, it is used in criminal procedure, it is used in forensic laboratories, and at last it is used in paternity testing, whether the child belongs to which parents. It is used by these applications. And all about the PCR. I think I hope you will all have enjoyed this lecture. And if you have any queries, then comment below. I will answer all of your questions. And at last, I thank you all and thank you.